The purpose of this module is to give you an introduction to using cloud-based computing as a means of generating a transcriptome assembly. So we'll start with this introduction of what is a transcriptome assembly, why might you need to generate one. So let's start with a hypothetical situation. Let's say you're a climate researcher, a biologist who wants to study and understand the effect of temperature changes on the black crappie reproductive cycles. So the way you might go about doing that would be to collect samples of the germ cells, sperm and eggs, and assess what's happening differently at different temperatures as these fish are trying to reproduce. Standard mechanism for standard tool of getting at the changes in samples at a molecular level is to look at the gene expression, to look at which genes are making transcripts, and how they are changing in response to the conditions. Our standard tool for doing this is what we call a transcriptome profile. Just as a basic reminder, the genes of an organism are encoded into the genomic DNA, and their activation or expression, as we call it, happens by a protein complex binding in and making copies of one strand of that DNA into RNA. A bunch of processing happens, but that picture of the RNA is the first level response of what is happening in the environment or in the developmental state of that sample. The transcriptome of an organism is, by definition, all of the RNA transcripts that can be made, that are encoded across all the genes within that organism's DNA. The differences among the different cells, the different organs, the different tissue types of an organism is which genes are turned on and which genes are turned off within that cellular sample. And finally, the transcriptome profile, or expression profile as it is sometimes called, is a measurement of each of the transcripts that are present within a sample, or at least our best estimation of all of the transcripts that are present within a sample. In the end, what we are trying to get at is some sort of a picture of what the changes are in the molecular response of a sample based upon some sort of perturbation. So this is a graphic representation of how you might look at the transcriptome profiles after they're done. In this picture, each column is one sample. Each row is one specific gene or one specific transcript sometimes. And the color represents the relative expression of that gene in this sample compared to all of the other samples. And you can see that there are dendrograms on both the bottom and on the right here, and those are the result of a hierarchical clustering, which represents the similarity of each of these things. And so from looking at something like this, we might identify a group of genes that are different in one set of samples compared to all of the others. So the fundamental idea of a transcriptome profile is that you can get a picture of the activity or state of that sample by a snapshot, a profile at one point in time of what's going on with those molecules. And as it says in a transcriptome, we're talking about the RNA transcripts. We believe that perturbations to the system will induce systemic responses that are reproducible as well. And finally, that comparison of these profiles between samples can either delineate the mechanisms or pathways that are involved in the response to that, and also to identify and classify the related samples, referring to the two different dendrograms we saw on the previous slide. So our standard tool for doing this is RNA sequencing. We're very good at generating nucleic acid sequencing. It's a technology that's been with us for over 50 years now, but especially in the last 10 to 15 years, it has really ramped up in our ability to generate a lot of it. So sequencing generally still, there are exceptions to this, but mostly it's still fragmentary. And so this is just a cartoon of the way this is generated. You have the cellular RNA, which is turned into DNA, cDNA, fragmented, a bunch of adapters and are added onto it. That's what's shown here as the colored sequences. The black part remains the part of the original transcript that we want to sequence. And finally, we end up with either single end or paired end sequence. But the critical thing here is that we only get this small piece of sequence from each of the original transcripts. And to put numbers on that, In short read sequencing, generally we're looking at 75 to 150 nucleotides, whereas the transcripts from which they're generated are usually hundreds or thousands or even tens of thousands of bases long. A critical step in interpreting and in building the answers that you need is the mapping of these reads onto the transcripts. And as it says here, in that searching, in that alignment or pseudo-alignment, you have the queries of each of the individual read sequences, and you've got the transcript sequences as the target. That's the point of our whole discussion here, is the target. If you're working in a common model organism, here we have the mouse, the zebrafish, the nematode, the fruit fly, you can see that a lot of the work has already been done for you. These are drawn from the current version of Ensemble, and there are 145,000 known transcripts in the mouse, 60,000 in the nematode. These are almost certainly not complete, but they're close enough to complete that you can make the analysis of your RNA-seq on the basis that how it matches to these transcripts will give you a good enough picture to know what's going on. But what about our crappie? There's no genome project and there is no reference transcriptome. How do we get a transcriptome profile in these conditions? Well, as you might guess, the short answer is we can build it. We have the information. If the sequencing reads that we have, RNA sequencing is a very high throughput and a very standardly done procedure now. So we can get enough sequence. We can regenerate those sequences, and that's the process that we refer to as transcriptome assembly. Conceptually, this isn't the way it's actually done, but this is conceptually what happens. You have short reads, you find the overlaps between them, and you extend them to get the complete read. This would be if you had one target. The problem with RNA sequencing, one of the many things that makes it challenging, is you don't have one sequence you're trying to recreate. You have many sequences you're trying to recreate. So again, this is a conceptual picture of it. These are meant to be the fragments, the RNA sequences that were generated from an individual sample. And the colors here represent the originating transcript. The process of building a transcriptome assembly starts by first trying to do our best job of clustering them together into those reads that are most likely to have come from a common set of transcripts. And once these clusters are generated, then finally to try to assemble those reads into specific transcripts. As it says below, this is somewhat similar to the idea of having multiple jigsaw puzzles that you could mix together and then try to separate and make into a picture. There are some interesting analogies that we could go into further, but this is the basic idea. You have to separate the pieces and then try to come up with the best picture of what the original sequence was that it came from. Several things make this challenging. Probably right at the top is the fact that RNA transcripts are not all unique. The genome has created lots of duplicates. There are gene copies. There are paralogous genes due to duplication at the genomic level. There are pseudogenes, non-functional copies of genes that might still actually be making transcripts, even if they don't carry out the function. And these will have common sequences very frequently, and so that complicates separating these off into their own clusters. Repetitive elements, retroelements, DNA transposons, these very frequently end up in functional transcripts, and so they will be in the express transcriptome that you're trying to assemble. This again creates overlaps and makes it harder to separate the reads into their groups. We have a wide range of expression. The transcripts are not all expressed at the same level. They differ between cell types, but even within a single cell, you'll have several orders of magnitude difference of how many copies of that transcript are present. That's going to affect the amount of evidence we have to put that back together. And finally, sequencing is not perfect. So you're going to get sequences that have errors in them, sequences that have mistakes. As you try to assemble your transcriptome, this is going to create possible alternatives. These are going to be false alternatives, but the program doesn't know that until you get enough evidence to try to separate that out. So at a very high level, transcriptome assembly is 6 conceptual steps. You have the gathering and processing of the input data, the sequences. There's the generation of the transcript. Generally, what we're going to do is generate it multiple times so that we can then assess, bring these together, and refine the transcriptome. After that, we're going to want to annotate the transcriptome. A transcriptome by itself is just a group of sequences. By annotation, I mean trying to assign functionality to it, trying to identify the sequences in there that we recognize by comparison with other organisms. We're then going to use the transcriptome. The transcriptome that we use is as a target for expression measurements or things of that nature. It could be used for phylogenetic analysis, any number of things. And finally, there's going to be the displaying and the sharing of the results. We're going to focus primarily on the first 4 of these in this module, but it's worth understanding what all of these are. So the key points that we want to get across in these modules are the following. We want to give you a picture of transcriptome assembly. We also want to talk about how we will do this using workflow systems. Workflow systems are a tool that allows us to repeat the same sorts of analyses over and over again for robust and reproducible work. And finally, of course, we will talk about how to efficiently carry this out in a cloud computing environment. And so with that, I will just say that if your organism of interest looks like any of these, there's a good chance that you're not going to have the resources that you need. And so transcriptome assembly is one of the tools that you will use in order to get the information you need to analyze your expression data.